Welcome everyone to our first ESR webinar series. And I'm going to be doing the first part of my talk where I'm going to talk about the pathophysiology of diabetic bone disease, and then later Sam will take over from me. So I'm based at the University of Sheffield in the Department of Oncology and Metabolism, and I'm doing my PhD under the supervision of Professor Richard Easter. So this is going to be the outline for my talk today. I'm going to touch on diabetes and the fracture risk associated with it, the changes in bone turnover in people with diabetes, the consequences of low bone turnover, as well as the causes for this low bone turnover in diabetes. In a systematic review and meta-analysis that was led by Tatiana Vilasa from a group here in Sheffield, she found that for type 1 diabetes, the risk of having a hip fracture was 4.9 relative risk, whereas for type 2 diabetes, it was 1.3. If we look at the risk factors for diabetes, she and others have identified that having diabetes for a longer duration of time, having a poorer control of your diabetes with an increase in falls, especially due to increased insulin use and hypoglycemia, having a low BMI, as well as being female, all these interact with diabetes and increase the risk of having a hip fracture. Now, whenever we see an increase in the risk of fractures, we wonder whether it can be explained by bone mineral density. So here I'm showing you a summary of studies of BMD in type 1 and type 2 diabetes. The BMD here is expressed as a Z-score, which is the number of standard deviations below the average for a given age for the spine and hip. We can see that in type 1 diabetes, the Z-score is reduced, but only by a tiny amount. In type 2 diabetes, surprisingly, the bone mineral density is increased by 0.4 to 0.3 standard deviation units. So then when you think about how we can translate this and what the expected fracture outcomes would be from these changes in bone density, for type 1 diabetes, the reduction would be somewhere about 0.3 or so of the standard deviation, and that will cause a hip fracture risk of 1.4. But we have observed the hip fracture risk to be above 4.9-fold increase in our studies. For type 2 diabetes, we would expect a reduction in fracture risk because the BMD is higher in these people, but we observe an increase in fracture risk in type 2 as well. So we feel that bone density is just not explaining all of what we see with respect to fracture risk. Now, I'm not saying that bone density does not relate to fracture risk in diabetes. In this study here from Anne Schwartz, she illustrates that lower the femoral neck BMD, higher the risk of having a hip fracture here on the left and non-spine fracture on the right. What's happening in diabetes is that for a given level of fracture risk, the bone mineral density is higher in diabetes, whether it's a hip fracture or a non-vertebral fracture. So when we try to understand the strength of the bone and the likelihood of it fracturing, we need to think beyond bone density, and we also need to be thinking about bone quality. In type 1 and type 2 diabetes, it is quite likely that there's a big alteration in the bone quality. So what we mean by bone quality is it's comprised of 4 main characteristics, which is the bone microarchitecture, bone turnover, mineralization of the bone tissue, and the accumulation of microdamage and development of microcracks. In my talk further on, I'm going to be focusing on bone turnover, and this bone turnover is actually found to be related to these other facets such as microarchitecture, mineralization, and microdamage. I'm not going to spend too much time on what the basics of the bone turnover markers are and the different bone formation and bone resorption markers today due to the lack of time. But in my talk, you'll notice that further on, I focus more on P1MT, which is a bone formation marker, and CTX, the bone resorption marker, as these are the markers most reliable in measurement and being proposed by the International Osteoporosis Foundation and IFCC. So here is a typical study from a group in New York showing what happens in type 2 diabetes. You can see that the P1MP is lower than in controls. Bone alkaline phosphatase is slightly higher, which may be due to the fact that it shares the same gene product with liver alkaline phosphatase. Liver alkaline phosphatase and bone alkaline phosphatase, the only difference between them is in their post-translational modification. And as a result, there could be quite a bit of overlap between both the ALP genes in the assay. Osteocalcine was also lower, and in this study, TRAP5B was not particularly different. And they also found a difference in CTX levels. The reason I'm showing you this table is because in the same study, they also assessed bone biopsies. So the question really arises is if the decrease in P1NP and CTX, is it also reflected in the bone biopsies? This shows what happens in the three envelopes of the bone, which is the cancellous bone, the endocortical part, and the intracortical part of the bone. And the variable that I found most important and useful to interpret bone turnover markers is the bone formation rate divided by the bone surface over here in the last column. You can see that for type 2 diabetes, there's about a 1/5 as that in controls. So there's quite a clear reduction in measuring bone turnover on the bone biopsies. And this is very much in sync with what is happening with P1NP and CTX. And in fact, the bone markers may be underestimating the decrease that is truly seen in people with diabetes. When we move on to what the consequences of the low bone turnover in people with diabetes would be, here I'm showing you a table from the Health ABC study, which was led by, one of the analysis was led by Nicola Napoli and Richard Easter. And the Health ABC study is a well-characterized study with older women in their 70s. If you start on the top here and we look at the bone marker for bone resorption, CTX, and this relationship to all clinical fractures, the darker circle represents the woman without fractures. Sorry, the darker circle represents women without diabetes. And so if you look at the classical appearance in people without diabetes, higher the CTX, higher the risk of fracturing. So here is a CTX increase of 20%, and so the consequence is a 10% increase in the risk for fractures. This is typical of what we would find in any other study where the bone resorption is linked to a higher fracture risk. Then when you look at the gray circle, we see that this isn't really true for diabetes. In fact, the opposite. Lower the CTX, higher the fracture risk. And already we are seeing something quite unusual, and this interaction was found to be significant. It wasn't very clear for a hip fracture or a spine fracture in this case. When we look at forearm fractures down at the bottom, we found something even more striking. Again, for non-diabetic women, higher the bone resorption marker, higher the fracture risk, and in diabetic women, lower the CTX, higher the levels of having a fracture risk. So there is definitely a supportive data from previous studies as well, where we probably need some more studies to validate this further. But it's really pointing us towards this direction of a low bone turnover, which is quite possibly having an effect in the increase in fracture risk that goes along with diabetes. So now, when we think about people with type 2 diabetes to have a low bone turnover, and when we look at postmenopausal women, whenever we see low bone turnover, we see a lower bone loss rate. But in diabetes, the opposite seems to be happening. Here in this study done from Anne Schwartz's group, we see that the rate of bone loss at the femoral neck is faster than it is in people without diabetes. Starting off with a higher BMD, of course, because there's always the case in type 2 diabetes where the BMD is higher than normal, but the rate of bone loss seems to be much faster, and yet they have a low bone turnover. And this seems to be quite interesting. In this study here, which was led by Serge Ferrari's group, we wanted to find out whether if women with osteoporosis and diabetes, did they have the same response to denosumab treatment as people without diabetes? And denosumab is the most powerful anti-resorptive therapy for osteoporosis. And over here in the FREEDOM trial, it is one of the biggest trials conducted with this drug. They found something quite interesting. Where not for vertebral fractures, where vertebral fractures prevented in diabetes, just like in non-diabetics, but for non-vertebral fractures, there was an increase in the non-vertebral fracture risk. And the fractures which were particularly predominant were in the radius and the ribs, as I've circled over here. The radius has found to be most important both in the Napoli paper as well as Ferrari's study as being possibly associated with particularly low bone turnover. Another point I'd like to touch upon here is one of the functions of remodeling is to remove micro cracks and micro damage in the bone. And here in this image with fusion red staining of the cortical bone, you can see two cracks, and the idea is that these micro cracks can propagate and cause a stress fracture. Moving on to what the consequences of the lower bone turnover would be, I'm going to be commenting more on hypoglycemia, accumulation of advanced glycation end products, and microRNAs. Speaking about hypoglycemia, do hypoglycemic cells actually have an effect on bone turnover? Here I'm showing you the relationship between non-fasting glucose levels and bone turnover markers. You can see that all the 4 bone turnover markers mentioned here have negative relationships with glucose. This makes you wonder if glucose itself is causing the changes in bone turnover, and if this effect occurs, is it actually a direct effect or an indirect effect of it occurring? The evidence for the direct relationship between bone turnover and glucose comes from this study here, where they have osteoclasts which are studied in culture. Here, the osteoclasts were stimulated with rank ligands and also incubated with either D-glucose, which is the normal glucose that is present in our bodies, or L-glucose, which isn't usually found. But you can see that it is only D-glucose which is inhibiting the osteoclast activity, with a picture shown here on the left, where the osteoclasts are inhibited. So in the study, actually, there were quite high levels of glucose being used, which was 25 millimoles. So it's quite interesting that perhaps the glucose itself might have an inhibitory effect on bone resorption. Next, talking about ages, I'm just going to quickly touch upon what they are. When proteins or other molecules react with glucose, various changes can happen, and these actually occur as slow reactions. They accumulate over time and do not involve the activity of enzymes. Here on the top, you can see as glycation occurs, the glucose interacts with the amino group of the protein. And one of the most common and important ages that occurs in this process is called CML, which is N-carboxymethyl lysine. This CML binds to its classic receptor called RAGE, the receptor of ages. And as a result of this interaction, that causes an increase in cellular activation and the release of pro-inflammatory cytokines, increased release of growth factors, apoptosis, oxidative stress, and the production of reactive oxygen species. This might contribute to diabetic complications and may also affect bone. The other types of ages function occurs via cross-linking, and pentosidine is an example of this kind of ages. The cross-links occur between one collagen molecule and the adjacent collagen molecule. This occurs as a result of formation of pentosidine, as I mentioned earlier, and it could lead to increased fibrosis, alter tissue stiffness, and might also affect the quality of the bone itself. In this study here by Anne Schwartz, she shows that in diabetes, the fractures are more common in patients with higher levels of pentosidine. And this is true for clinical fractures, but it wasn't true for vertebral deformity so much. And also it is related to the rate of bone loss. Higher the rate of bone loss was seen in patients with higher pentosidine. So this actually helps us form an interesting link between ages, bone loss, and fracture risk. This theory that ages might also be working via altering bone turnover, the evidence for this comes from a review where they explain how ages may relate to bone, and the authors propose that hypoglycemia, which results in the accumulation of ages, and the ages bind to the RAGE receptor, and they have a number of effects. In the long term, the accumulation of this leads to decrease in proliferation and the formation of osteoblasts and an increase in osteoclasts in this model. In prior data, it has also been shown that more the accumulation of ages, lesser the osteoclastic activity and bone resorption. So it is quite possible that ages are mediating some of these effects that we've already seen in bone turnover, but this area requires more understanding and validation. Talking about CML, this data here is from an abstract that was submitted to the ASBMR in 2020, where they found a relationship between CML and fracture risk in the Health ABC study. CML in this study was measured using ELISA. They found that for every standard deviation increase in CML in serum, there was a 1.45 factor association with fracture risk, and it was also associated with prevalent fractures. They found an association with clinical fractures and borderline vertebral fractures. So it kind of goes in the direction that both CML as well as pentosidine seem to be associated with increased fractures in people with diabetes. Now switching over a little bit to microRNAs. MicroRNAs are small, highly conserved, non-coding RNA molecules which are involved in the process of regulation of gene expression. I'm going to show you two studies here. This first study is where increased serum levels of microRNA 148A and 21,5P were found in people with type 1 diabetes, and these microRNAs were found to be correlated with markers of bone strength and metabolism. The data of this study comes from a well-characterized type 1 diabetes study cohort, where characterization was done for a specific panel of bone metabolic markers and bone physical parameters. In this study, they measured 6 microRNAs in serum, and they found an increase in expression levels of the microRNAs for microRNA 148A and 21,5P. If you look at the graphs down at the bottom, they found a significant negative association between microRNA 148A and bone mineral density at the total body level, which forms a potential link between bone strength and this particular microRNA. They also found a positive correlation between 148A and PTH, which forms a possible link between the microRNA and bone metabolism. Switching over to microRNA 21,5P, they found a significant correlation between this microRNA and bone mineral content at the femur. And both these microRNAs provide good evidence for the modulation of bone strength in people with diabetes. If you look at the heat map on the side here, where they had done a bioinformatic analysis using a KEGG pathway analysis for these two microRNAs, 148A and 21,5P, and their target genes, they showed that FOXO signaling pathway and the TGF-beta signaling pathway are the main two pathways that come up. And these two pathways are found to be significant with these microRNAs and important roles in bone metabolism and remodeling processes. Here is another study where they look at a bunch of microRNAs, about 10 microRNAs from the Icelandic age study, which contains postmenopausal women with type 2 diabetes with and without incident fractures. In this study, the 10 microRNAs that were measured or studied are listed on the table on the side. And what they wanted to see whether these microRNAs chosen had any associations with incident fragility fractures. So in order to identify this, they performed a Cox regression analysis of all these 10 microRNAs together. And based on the regression coefficient and the p-values registered with the model that was used for the study, they found microRNA 1915P, 203A, 315P, and 181C to be more significantly associated and promising microRNAs for incident fracture identification. They then took these 4 microRNAs individually and tested them via Cox proportional hazard modeling separately, and they looked for the association between the baseline circulating levels of the microRNAs and the risk of an incident fracture occurring. In the unadjusted Cox proportional hazard models, circulating levels of microRNA 203A and 315P were significantly associated with a higher risk of incident fragility fractures. With further models that were used for the study, These two microRNAs, 203A and 315B, remain to be consistently significant. To sum up, I've tried to show you that diabetes is associated with an increase in fracture risk, and this is best explained by the changes occurring in bone quality. Bone turnover is decreased in people with diabetes, both type 1 as well as type 2 diabetes. And this low bone turnover is associated with an increase in bone loss and possibly an increase in fractures. And the causes of low bone turnover could include the accumulation of advanced glycation end products and microRNA changes in expression. I thank you all for your attention, and I'm happy to take your questions.
Thank you, Ankita, for the lovely presentation. I would say it's Sam's turn now, and then after both presentations, we have time to let people ask questions.
Yeah, okay.
Thank you very much.
Okay. You see my slides?
Yes, very well.
Fantastic, thanks. Okay, I'll begin. Sorry, you already introduced me at the beginning, right? Sorry. So my name's Sam, I'm ESR10. I'm based in Rotterdam, in Erasmus MC. This is in the Netherlands. My supervisors are Ling Oy and Professor Fernando Rivendineda. Today, I'll talk about the genetic overlap between type 2 diabetes and bone mineral density. And this is actually just one project that we're recently wrapping up. And this is a Mendelian randomization and statistical colocalization study. So this is just an overview of what I'll speak about today. In the introduction, I'll talk about why there's a need for more research in this topic. What is Mendelian randomization? I'll then move on to the methods and the study design in this project. Discuss with you through the results and summary of main findings. And then I'll move to the conclusions actually, and then I thought we can do the discussion as a group right at the end. We have a lot of time at the end, so we can discuss together what the results mean. So Ankita just brilliantly gave us an explanation of everything, so I don't have to go through much. But what I will say is there's a need for more research because currently there's no kind of evidence-based treatment guidelines on how to manage patients with type 2 diabetes who might be at risk for, it shouldn't be osteoporotic fractures, they should just be fractures. This is a typo. Is it better to give them anti-resorbative drugs or anabolic bone-forming drugs? This is kind of unclear. We have ad hoc analysis of randomized control trials, but obviously this is not ideal. And I think further, basically, on a basic research level, it's really interesting to study, right? Because it gives us an opportunity to study mechanisms of bone fragility that are not explained by bone mineral density. So I think mechanistically, this is really interesting on a basic research level. But today I'll talk about Mendelian randomization and causal inferences. So I'll start by talking about how can we gain causal inferences and what are causal inferences. So if we take this example here, let's say we want to see if smoking increases your risk of mortality. So how long you live, basically. How might we do this? How might we find this out? So what we can do is we can look in the general population, and we can look at individuals that smoke and those that don't smoke. And we can basically see how long does each group live for. And if those individuals that don't smoke live longer lives, then we can gain some inferences, right? And maybe conclude that smoking leads to a shorter lifespan. This is what's done in observational epidemiological studies. But of course, it's not really this simple because those people that smoke, they also tend to drink more alcohol. They have more unhealthy diets. They undertake more risk-taking behaviors, for example. And all of these things also are associated with mortality. So we call these confounding factors. So this raises the question, right? Is smoking really leading to a shorter lifespan, or is it just they share these confounding factors which also reduce your lifespan? So what they do in observational studies is they control for these factors, right? They put these in the model and they control for them, but this is not always possible, right? You can't always really measure these accurately, these confounding factors. And also you can't always think of every confounding factor that affects the two things that you're interested in. This is obviously not ideal. But actually, in observational studies, that's why they don't claim to make observe causal associations. They just claim to see evidence of significant evidence of an association. They're not claiming causality. So what can we do if we wanted to claim causality or gain some causal inferences? Some people say this is not possible at all. So we can never gain causal inferences. But I think this is more of a kind of philosophical question about, about semantics, really. So what is your definition of causality? If you have a very strict definition, then of course, nothing's going to satisfy your definition. If you have a more practical definition, then you can definitely satisfy the assumptions for causality. So what definition should we use for causality? I think a good definition to use is what they use, kind of the clinical gold standard, which is randomized control trials. So this is seen as the gold standard for clinical practice. And in randomized control trials, we can take a group of individuals, randomly assign them into two groups, give some a placebo, some the treatment, and then any effect that we measure in the outcome, we know is due to the intervention that we cause and not confounding factors, because the confounding factors are randomized between the two individuals. So this is considered kind of some kind of causal inference that we can gain. And it's good enough for clinical practice. So I think it's a good approximation for us to make. So let's say we want to do this for our smoking example. We can take 2,000 healthy individuals. We can give half of them, placebo cigarettes, half of them real cigarettes, tell them to smoke 20 a day, and then tell them to do that for 20 years. And then we can see, how many are still alive by the end of the study. But of course, this is not really a real experiment or a non-ethical experiment to run. So in this case, we'd never be able to do a randomized control trial. So what can we do instead to gain some causal inferences? Well, luckily, genetics or nature has allowed us, has enrolled all of us in a randomized control trial from birth, which we can utilize. So we know that there's certain genetic factors that increase or decrease your risk of smoking. These are randomly assigned to you at birth. So we have a randomization of confounding factors. So we can actually use this to generate two groups in the same way we do as a randomized control trial. So we can do the same. We know the factors that increase, you have certain alleles that increase your risk of smoking, and those that don't or decrease your risk. You can split individuals based on that. The individuals will be randomized in terms of confounding factors. Same as a randomized control trial due to the law of segregation from Mendel. And then again, we can look at the life expectancy of the two groups. And in this way, we can gain some causal inferences about the effect of smoking on mortality. So in a nutshell, this is Mendelian randomization. It's called Mendelian randomization because of the randomization, right? In the same way as a randomized control trial, and Mendelian for Mendel and his three laws, which are utilized in the study design. So we can apply this to our topic, right? We're interested in how type 2 diabetes affects fractures and bone mineral density. So we're trying to evaluate this relationship here, independent of these confounding factors. So we can take genetic variants, which we know are associated with type 2 diabetes via GWAS studies. And we essentially test if these are also associated with bone mineral density or fractures. And if it's true, given some assumptions, so if the genetic variants are associated with bone mineral density, then we can assume there's a causal association between these two. This is actually a form of instrumental variable analysis, which is basically developed by economic statisticians a long time ago, but recently has been applied to genetics and epidemiology, because genetic variants really satisfy pretty well the assumptions for an instrumental variable, which is quite difficult to find. So what are these assumptions? Firstly, the instrumental variable, which is a genetic variant in our case, needs to be reliably associated with the exposure. So this is easily determined by GWAS studies. So this is an easy assumption. The next basically says that your instruments shouldn't be affected by confounding factors. This is also pretty easily established because your genetic variants are fixed at birth, right? So this is an easy assumption to give also. The third assumption is a bit more tricky, which is what most of the work goes into, evaluating and how you can get a lot of erroneous results. And this is that the instruments should only be associated with your outcome, so bone mineral density, through your exposure to type 2 diabetes. So it should follow a path like this. It can't go something like this. And this is especially true in our case, right, with type 2 diabetes and body mass index, because a lot of the genetic variants that affect type 2 diabetes also affect body mass index, which can also affect bone mineral density through mechanical loading. And this violates the assumption, right, and this can give you wrong results. So this is something you really need to be on the watch out for, which we will be in the study. So this is not a new idea, applying this to diabetes and bone. There's been a few papers before that have done this. So one paper from Brent Richards' group in McGill on the left, another from Mark McCarthy's group in Oxford on the right. And what Brent Richards' group did is look at the traditional association type 2 diabetes on bone mineral density. And they found evidence of a causal effect, or causal association between the two using Mendelian randomization. And then Mark McCarthy's group did the opposite, actually. They looked at, do the processes that lead to increased bone mineral density, do they also increase the risk of type 2 diabetes? So this is kind of a flipped association, something that's not as well studied. And they actually found, again, significant evidence of a causal association, which I think was quite surprising, to me anyway. But these studies aren't perfect. They have a lot of limitations. So we can definitely do some improvements on these studies. Firstly, the GWAS summary statistics, which are used to do the analysis, were already adjusted for BMI and weight. And this can introduce something called collider bias, which can lead to wrong results, essentially. We can apply updated methods, such as multivariable MR, to remove the effects of body mass index more effectively. And we can apply something called colocalization, and this can be used for two different purposes. First is to remove genetic variants that violate this independence assumption that we saw the third arrow on the last slide. And also you can highlight genomic loci for functional follow-up. I didn't have time to go into this, but essentially what this does is look at the genetic overlap of genetic variants between two traits. So in our case, that would be type 2 diabetes and bone mineral density. So this is what we plan to do in our methods. We plan to apply these improvements to the paper and see if the results remain consistent. So we did this for bone mineral density and type 2 diabetes in both directions, controlling for the effect of body mass index. And we also looked at the effect of type 2 diabetes on fracture risk, adjusting for the effects of bone mineral density. We obtained GWAS from the largest studies available. We used eBMD for the genetic variants for bone mineral density. This is estimated from Hill ultrasound. This is used in genetic studies because it characterizes really well the genetics of bone mineral density, and it has a really large sample size, almost 500,000 people. So this is why this is used instead of something DEXA derived. We also have fracture risk and BMI, and we performed multivariable Mendelian randomization, and we also did statistical colocalization, which again, I can't really speak so much about. So results, I'll give you a quick summary of the main results that we found. Here's the first slide. This is the results of the Mendelian randomization. Maybe there's a lot going on here, so I can go through it quite slowly. Here on the right, we have a forest plot. And this is essentially the results of a regression, really. With your diamonds is your effect size, or beta. And these confidence intervals or lines, these are your confidence intervals. If it passes this vertical threshold, you know that it's significant. So if we begin at the top, this is the effect of bone mineral density on type 2 diabetes risk. This is the number of genetic variants that are associated with bone mineral density we have here. And this is the p-value of basically what is the inverse variance weighted regression. So it's just a regression, basically, of the genetic variants. So what we see here is actually, really significant result. So this indicates that the processes that lead to increased bone mineral density also increase type 2 diabetes risk. And this also remains significant after adjustment for BMI. Even though we lose a lot of genetic variance and the p-value increases because of this, the effect estimates remain really consistent, which is a really good sign and something you should be looking for. We also further validated this in a DEXA-derived total body BMD. This has a lot lower number of genetic variants because it's a much smaller study. But again, the effect estimate remains really consistent and is still significant. So this is quite surprising, actually. This is something that we didn't expect. I expected to kind of disprove the results from Mark McCarthy's group because I thought there's some errors in the way they've done the analysis or some kind of biases. But in fact, we reconfirmed the associations and the effect estimates are really consistent with their study also. So this is quite surprising. Next, we looked at the effect of type 2 diabetes on fracture risk. So this is obviously what we're all studying. The first study is unadjusted, the next is adjusted by BMD. The unadjusted, we see basically a borderline negative association, but then this is removed after adjustment for bone mineral density. This was expected because essentially fracture is not a not a robust genetic study has been done on fractures really. The largest fracture GWAS, for example, is something like 1 million people, and they discover only 12 or 14 genetic variants. In comparison, type 2 diabetes, which is around 800,000 or even less, has 300 to 400 genetic variants. So fractures is really not well characterized genetically, and the genetic variants we do see in fractures are really explained by bone mineral density. So when we adjust for bone mineral density, we really lose all of the association. And again, this is really expected because in type 2 diabetes, bone mineral density is really not explaining much of the fracture risk in those patients, although explaining some, of course. So this was actually expected. And lastly, this is the expected direction around the effects of type 2 diabetes on bone mineral density. This is a really robust association. It's seen epidemiologically as well as from Mendelian randomization studies and genetic studies. So first, again, we replicate the association with a larger sample size. Once we adjust for the effects of BMI, the effect remains again really consistent and really significant. So this indicates still that the effect of type 2 diabetes on bone mineral density is causally associated independent of body mass index. And lastly, one last analysis we performed was the effect of fasting insulin levels on bone mineral density. So there's a popular hypothesis that the increased bone mineral density in type 2 diabetics is driven by the anabolic effect of insulin on bone. And of course, type 2 diabetics have higher insulin levels. So we performed this using 55 genetic variants from fasting insulin GWAS. And we see a significant result. So the higher your fasting insulin levels, the more bone you seem to form. The confidence intervals are really large for this, but this is because the number of genetic variants is quite low, 55. But the effect estimate is really large. So it's 10 times that of type 2 diabetes. This also makes some sense because when we're looking at genetic variants and Mendelian randomization, we're looking at lifetime effects. And of course, type 2 diabetes occurs really late in life. Most people 50 or 50 plus, whereas the insulin levels are going to affect your bone throughout your whole life. So it makes a lot more sense that the effect estimate is much larger. But this is also quite promising as a potential explanation. I'll briefly speak a little bit about the colocalization results. I couldn't really explain a lot about the theory behind it before, so I'll just briefly mention something. All we're essentially doing with colocalization is looking if genetic variants from one trait, type 2 diabetes, overlap with another trait, BMD. So we did this across 243 type 2 diabetes loci, and we found 29 overlap with a high degree of correlation between the SNPs in those loci. This is what we see in this plot here. Sorry. And we identified CCND2 as a potential gene for further follow-up. So the variants that overlap between type 2 diabetes and bone mineral density are really close to this gene. I mean, within 200 base pairs, which is really, really close. They have almost perfect overlap, meaning it's the same genetic variant affecting both traits. And they also have a really large effect on both bone mineral density and type 2 diabetes. And this gene is really unwell characterized, to be honest. There's not much known about it, especially in the context of bone mineral density and type 2 diabetes. So this really indicates an underlying biological mechanism that's shared between the two traits, and this gene seems to be responsible. In comparison, we can look at something different like TCF7L2. So this is a transcription factor for the Wnt pathway, a really important transcription factor actually. And it also happens to be the largest genetic variant associated to type 2 diabetes. So this is the p-value, the minus log 10 p-value, and this is minus 400. It's really, really strong signal for type 2 diabetes. So this is often cited as evidence for the shared biology in the Wnt pathway between type 2 diabetes and bone mineral density. If we check if this genetic variant overlaps with variants in the bone mineral density GWAS, we see actually it doesn't overlap with the top hit from bone mineral density GWAS in comparison to BMI as a comparison where it perfectly overlaps. So this indicates that while they both affect this same transcription factor, they have slightly distinct biological mechanisms. And since it's a transcription factor, I mean, it affects lots of different genes. So even slightly different mutations that affect this transcription factor probably affect lots of different genes in different ways in a much further downstream pathway. So while this provides some evidence, right, they both affect the same transcription factor, it seems that they affect it in different ways. So there's definitely some overlap in the Wnt pathway, but it might not be the exact same biological mechanism. So now I'll move on to the conclusions, and like I said, maybe we can have the discussion at the end. So the key results, we observe a bidirectional causal relationship between bone mineral density and type 2 diabetes. And this is even after adjustment for BMI and removing genetic variants that violate instrumental variable assumptions via colocalization. We observe that increased insulin levels are causally associated to increased BMD. After adjustment for bone mineral density, we observe no causal association between type 2 diabetes and fractures. We observe overlap at 29 loci between type 2 diabetes and bone mineral density. And at the TCF-L2 locus, we observe distinct causal variants that are affecting the gene in different pathways, which shows that permutations in this transcription factor affect glycemic control and bone mineral density through slightly different biological mechanisms. And to conclude, we suggest that higher bone mineral density, when considered in the context of age and lifestyle factors, of course, these patients should be flagged as being at risk for type 2 diabetes and can be followed up with glucose tests. And lastly, we identified TCND2 as a potential target to evaluate the shared biological relationships between type 2 diabetes and bone mineral density.